Dear viewers, greetings. In this video, we are going to see about the gram-positive rod Clostridium tetani. Before going to the general characteristics of Clostridium tetani, I like to give a small introduction about the Clostridium species. The collection of all anaerobic gram-positive rods capable of forming endospores were placed in the genus Clostridium. The genus Clostridium species was defined by four properties. The first property is presence of endospores. The second property is strict anaerobic metabolism. The third property is inability to reduce sulfate to sulfide. And finally, the fourth property is gram-positive cell wall structure. More than 200 species of Clostridium have been defined currently. The organism Clostridium species are ubiquitous in soil, water and sewage and are the part of normal microflora in gastrointestinal tract of animals and humans. Most Clostridia are harmless and saprophytes in nature because they are living in dead and decaying organic matters. Some of the Clostridium species are well recognized human pathogens with a clearly documented history of causing disease such as tetanus caused by Clostridium tetani, botulism caused by Clostridium botulinum, Clostridium barretti and Clostridium butyricum, myonecrosis or gas gangrene, that is the infection of muscles caused by Clostridium perfringens, Clostridium novi, Clostridium septicum and Clostridium histolyticum, and finally diarrhea and colitis caused by Clostridium perfringens and Clostridium difficile. The remarkable capacity of Clostridia to cause disease is attributed to their ability to survive adverse environmental conditions through spore formation, rapid growth in a nutritionally enriched and oxygen-deprived environment, and production of numerous histolytic toxins, enterotoxins, and neurotoxins. General characteristics of Clostridium tetani. Clostridium tetani is a gram-positive drumstick-shaped bacilli arranged in form of chains and motile in nature due to the presence of peritrichous flagella and its size ranging from 0.5 micrometer to 2.5 micrometer, and it belongs to the family Lactinospiraceae. Capsules are absent and the endospores are present with a terminal arrangement that gives a tennis racket appearance. Regarding the respiration, the mode of respiration is anaerobic respiration. Optimum temperature for the cultivation of Clostridium tetani is 33 degrees Celsius to 37 degrees Celsius. Optimum pH is 5.9 to 9.5. In habitat, the spores of Clostridium tetani are present in soil and gastrointestinal tract of humans and animals. Regarding the discovery, the Clostridium tetani was discovered by the German scientist Arthur Nicolaier in the year 1884. Disease transmission of Clostridium tetani. The Clostridium tetani are easily available in the soil and dust particles, which is easily enters through the wound area and infect the humans by means of blood. Virulence factors of Clostridium tetani. Virulence factors are the factors which are responsible for causing the disease in human beings. And the term virulence was introduced by the scientist Louis Pasteur. And the term virulence means the ability of the microorganisms to cause an infection in human beings. Clostridium tetani have the ability to produce two types of toxins as a virulence factors. The first one is tetanolysin and the second one is tetanospasmin. The tetanolysin is a oxygen-labile hemolysin and the tetanospasmin is a heat-labile neurotoxin. And the toxin tetanospasmin was produced during the stationary phase of the Clostridium tetani growth. The incubation period for Clostridium tetani is 4 days to 3 weeks and the infective dose required for causing an infection is variable. Pathogenesis of Clostridium tetani. The spores of Clostridium tetani enters its host through open wounds or burns where many cells are dead. The wound or burns provides the necessary environment for the Clostridium tetani to begin to spread its virulence factors. In the presence of anaerobic condition, the spores can germinate and the spores produce toxins like tetanolysine and tetanospasmin that are harmful to the human nervous system. The toxin tetanospasmin travels through the body via the nervous system and reaches the spinal cord. From spinal cord, the toxin relay begins to attack the body. Finally, the toxin tetanospasmin interferes with the neurotransmitters and blocks message to the brain. This leads to the spasms or muscle contraction or tightening or shortening of the muscles. In severe cases, individuals can experience severe seizures or changes in the brain electrical activity. Clostridium tetani results in 4 types of tetanus. The first type of tetanus is generalized tetanus. The second type of tetanus is localized tetanus. The third type of tetanus is cephalic tetanus. And the fourth type of tetanus is neonatal tetanus. The first clinical disease caused by the Clostridium tetani is generalized tetanus. Generalized tetanus is the most common form of tetanus, and the involvement of the master muscles is the presenting sign in the most patients. Master muscles are the facial muscles that plays a major role in chewing of food. The characteristic sardonic smile, that is looking like smiling, that results from the sustained contraction of the facial muscle is known as rhizus sardonicus. Other early signs are drooling or saliva has come out from the mouth, sweating, irritating, and spasms or muscle contraction or tightening or shortening of the muscles. The autonomic nervous system is involved in patients with more severe disease. The autonomic nervous system regulates the functions of the internal organs of the human body, and the signs and symptoms includes cardiac arrhythmias, that is improper beating of heart, fluctuations in blood pressures, profound sweating or abnormal excessive sweating, and finally dehydration. The second clinical disease caused by the Clostridium tetani is localized tetanus. The localized tetanus is very uncommon, and here the spasms restricted to the localized area of the primary infection. And in this condition, patient with this clinical type experience muscle rigidity close to the site of injury. The third clinical disease caused by the Clostridium tetani is cephalic tetanus. Cephalic tetanus is a form of localized disease that affects cranial nerves. Cranial nerves are the 12 pairs of nerves which arise directly from the brain and pass through separate apertures in the skull. Cephalic tetanus can happen after the ear infection or head injuries. And the cephalic tetanus affects the cranial nerves, so it can affect the muscles in face, eyelid, tongue, lips, etc. The fourth and final clinical disease caused by the Clostridium tetani is neonatal tetanus or tetanus neonatorum. Neonatal tetanus form a generalized tetanus that occurs in newborn infants, and it usually happens when the umbilical cord is cut with an unsterile instrument, and finally the mortality in infant is very high and it exceeds 90%. Laboratory diagnosis of Clostridium tetani. First diagnostic method is microscopic examination. Under gram staining, the Clostridium tetani was observed as violet colored gram-positive drumstick-shaped rods. In motility test, the cells are motile due to the presence of peritrichous flagella. In endospore staining, green color endospores with terminal spherical spores were observed, and red color vegetative cells are also observed. This image shows the gram staining of Clostridium tetani, and here the Clostridium tetani is observed as drumstick shaped gram positive rods. And this image shows the endospore staining of Clostridium tetani, and here the endospores are observed in green color and the vegetative cells are observed as red color. And next, the colony morphology on culture medium. In nutrient agar, the Clostridium tetani growth appears as a swarming layer throughout the medium. The layer is so thin and transparent. In blood agar, the growth of Clostridium tetani appears as swarming layer over the surface of the medium. Initially, it shows the alpha hemolytic colonies followed by the beta hemolysis, which is due to the action of the tetanolysin or the hemolysin toxin produced by Clostridium tetani. These colonies are flat, translucent and grayish with an irregular margin. In McConkey agar, green fluorescent colored non-lactose fermenting colonies were observed. The selective medium for the cultivation of the Clostridium tetani is Robertson's cooked meat broth. Clostridium tetani grow well, produce turbidity, and the meat is not digested but may turn black, especially on prolonged incubation. It also produces gas and turns pH of the medium acidic. Biochemical test for Clostridium tetani identification. Catalase test negative, oxidase test negative, urease test negative, indole test variable, MR test or methyl red test negative, VP test or Voges-Perot score test negative, citrate utilization test negative, starch hydrolysis negative, casein hydrolysis negative, gelatin hydrolysis positive, nitrate reduction negative, hydrogen sulfide production positive, and finally DNase production positive. Finally, treatment and prevention of Clostridium tetani infection. The treatment requires debridement or removal of damaged tissues, antibiotic therapy with penicillin and metronidazole, passive immunization with antitoxin globulin, and vaccination with tetanus toxin, which was prepared from the toxin tetanospasmin. And regarding the prevention through the use of vaccination, it consists of 3 doses of tetanus toxoids followed by booster doses for every 10 years. And dear viewers, that's all about the bacteria Clostridium tetani. Thank you for your support. Thank you.